How's everyone going? It's so fantastic to kind of see some of these talks from this morning. I think personally for me, there's nothing really more gratifying than kind of seeing the things that people are building with the tools, with the kind of from within the Nextflow community. I get the really, I guess, kind of the lucky opportunity to present a lot of the work that we've been doing for the last 6 months or so as we sort of spread out these product releases. There's actually really a lot of things, and there's really a lot of people's work that I'm going to be presenting here today. But before I go into all those shiny new things, I want to start with a little bit of an observation and something that we've sort of been seeing more recently. Now, I'm by no means a software engineer. I am very much an armchair observer on Twitter and have kind of seen debates around different tech choices, different programming languages, different things that constantly go on. I think it's very clear that the pace of change within software development has really changed. Now it's very a lot of like adoption of new technologies. I guess you could chalk this up to engineers really liking to build stuff and particularly building things themselves. I know as both a startup CEO as well as sort of having done sales in the early part of Seqera, you see there's nothing more appealing to engineers than build it yourself. And in some ways, this has actually been really kind of positive. If we think about a lot of the kind of tooling and that has been built for developers, it's really benefited sort of software generally and has improved a lot of software development over time. We think about the things of like code generation, which is just how you write code. IDEs like VS Code have become really, really prominent. The new generation of code generators that are coming from LLMs, it's like very exciting. And the ability to kind of really build both kind of like core code, but also even scientific code more broadly. Source code management is really key. The use of Git in particular and the ability for people to kind of all come together on that same code base has really enabled a lot of improvements in terms of the way that we develop things. The packaging, if we think about kind of Docker and containers, this ability to take software, to publish it, and then for anyone to kind of be able to use it in this way. I think it's actually 10 years ago, sort of recently, since Paolo sort of first showed me the talk of Solomon Hykes. This is really from the original Docker talk that was done, I think it was in PyCon in 2013. And really kind of from that idea, it's just really kind of taken over the world of delivery of software. And so many of the things that are run on that today. Cloud is kind of a more obvious basic one. We all think about the elasticity and almost the infinite resources that we can pull from the cloud, if you're willing to pay for it, of course. The storage and the compute which comes with that, and then kind of all the additional services which have been built on top of that, making things easier, things like batch, etc. As well as the testing, and I think the unit testing all the way from individual snippets of code that we have, and some of this is coming through an NF core, some of the things like the full-size integration tests, which are just absolutely invaluable for really understanding exactly what changes are you making in the code, and sometimes the small little things that you affect can end up with hugely different results. So being able to have a full grasp of that is really key. And then finally onto Paula's favorite piece of software, Jira. The works of like project management and the like has become really key just to be able to coordinate larger software teams as we do that. But given all these benefits and given that all of us spend probably the vast majority of our day looking at computer screens, why haven't all of scientists benefited from this necessarily? I think for us, it's really believe that it's time for scientists and for science more generally to take advantage of the benefits of modern software tooling. I guess I'd ask, how can everyone in this room help empower those scientists with these tools? How can we kind of make them more relevant? Because for a group of us, it's made it work so much easier, but how can we apply that more generally to all of science? I guess it's about removing barriers in some way. It's reducing hurdles, and it's kind of making these tools relevant and making kind of open science a reality for everyone. Maybe before we answer how, and I'm hoping that we can kind of show you a little bit of how later on, thinking about the why. Like, why would we want to do this? Why would we want scientists to use modern software tooling? And for us, we really believe that there's better science is done with better software. Back in 2016, I remember that I was in a pizza shop with Paolo. There was no pineapple involved, thankfully, but we was thinking about the original Nextflow publication, and at the time, we were looking at a genome annotation pipeline, very complex thing that was coming from Sanger, and it was really the first pipeline that we tested in terms of understanding what happens if you could containerize all the pieces versus just using the pipeline itself. I spent something like 3 weeks installing every single piece of software on the cluster and ran it in that way. Finally got to the result and was like, yes, we can really do this. And then realized the results were different. Like, there was just subtle differences in between what we were doing, even though all of the versions were the same. And it kind of got to the point where I realized that, like, how can we be sure of anything in terms of reproducibility with that? And it's really just one example where using software tooling, in this case containers for reproducibility or Git for tracking code, really results in better science. This is just kind of really one example of it. I think, really we believe, like, when science comes together with software, amazing things happen. And Seqera is really the convergence of those things. It's really where science meets software. The development of Git enabled the collaboration of experts to come together. The technology really helps people come together and unleash the magic of people. And that's really what NF Core is a lot at its heart, is the people being able to come and build together. If we also think of some of the other technologies sort of listed here, and these are by no means like necessarily the main ones, but we think about cloud. It's a 20-person biotech startup in Boston is not going to go and spin up their own, look after their own racks anymore, the ability to kind of pull into cloud like that and build on top of things. The pains of manually uploading, downloading, sharing data, we really want to make those things a thing of the past. And that's really what our mission has been on for the last 10 years or so. It's that mission of advancing science for everyone through software, really bringing people together, and they're really making sure that the software is allowing us to do better science. Okay. So I've talked a lot about software so far, and that's kind of one thing. And technology, in a way, is kind of cool. Like, we all like technology, we all like to do things, but I don't think any of us in this room wants to learn Kubernetes so that we can sell more clicks or anything like this. We're really here to advance science and to do that. And for us, how that means kind of advancing science, we've got a couple of ways that we want to do that, when we want to help to achieve that. One of them means being able to deliver almost like an end-to-end solution for scientists. It means from the development of code, connecting in with data resources that you have, being able to spin up computer environments, interactive analysis, kind of that full reporting, and just manage that full data analysis lifecycle. And it's when we think about a kind of an end-to-end solution for us. As it relates to everyone, I think that's been really clear from the talks that we've seen this morning, and particularly what Gigi was mentioning before, the ability to kind of do this in an open science community. We think that in this way, because of what software enables, it allows us to stand on the shoulders of those amongst us. We can all learn from each other so quickly now and collaborate so fast. The Nextflow community in itself it's got some of the greatest minds of our time. And we can tap into those resources, collaborate, and learn from each other when we do that. Open science is more than open source, though. Open source is just a kind of a license. Whereas kind of open science is much more of a movement, but it's a movement which is kind of built on open source. And this ability is to build on open source and then to have the kind of technology enable the science in itself. If we think about those three pillars, they're the things that we use to orientate ourselves at Seqera. They're the things that get us excited, the things that we can kind of build towards. And so far, the way that we kind of think about that in a way is three things. So I know it's easy to remember things in threes. It's also easier to remember single words in threes. So for us, that is around kind of build, discover, and scale. Build with open source tools, things like Nextflow. Be able to discover resources through a community of experts, through things like NF Core, as well as kind of scale an end-to-end platform. And to date, that's kind of looked a little bit like this. We have core offerings of Nextflow, MultiQC, Wave. So these are kind of core tools that have been used. I've been building a lot on some of the forums and communication around documentation and NF Core, and then kind of scaling things out. But today I've got a whole bunch of sort of exciting new things that are also related to all of these. And I kind of want to kind of jump now into some of those pieces. Okay, onto the new stuff. If we think about some of the foundational resources that exist in kind of the world of software, you often think of things like GitHub. And GitHub is the home of code and something, or the home of open source code for many people. It's driven by an underlying technology, Git, which has been able to kind of drive a lot of that. More recently, there's some like new examples of these kind of open resources which have become really powerful. You're familiar with like Hugging Face. And in particular, that's like really the home of ML and AI models. It's really driven by an open source library which anyone can just pull into their Python and then kind of run these models. And there's like 1 million models there. Our real view of Seqera is to be the home of open science. It's this home of open science driven by the kind of resources and driven by the technology that exists there. I'm going to walk you through an example today, and just as a single example that we can use, hopefully kind of drive through some of those points and kind of as well kind of show some of the new things that exist there. So if we think about a single whole genome sequence analysis here, this is like one that I, sort of an exercise that I went through the last couple of weeks to kind of look into this. This is not trivial in terms of the amount of software that's involved, different libraries, different command line tools, all of the different modules in a Nextflow pipeline or an NF Core pipeline in this case, the different data sets. Given our mission is to advance science for everyone, we wanted to like really make that basic scientific software better for everyone. And for us, one of the core, let's say, primitives of that is containers. And particularly like containers of scientific software. How can we make those containers of scientific software available and easier for a broader audience to use? Containers solve the portability, the reproducibility piece, but they don't necessarily solve themselves. So today we are introducing Seqera containers, and this is something which is available today on Seqera.io. What this enables you to do is build from your most popular Conda packages, bioconda packages, Python packages, and what happens is all of those containers themselves get built, in this case using Wave, and they're stored up in AWS and then used by Cloudflare. Provides this kind of high availability, low latency, wherever you're running your compute. Some of the origins of this date a little bit back to when we had Wave, which is this concept of instead of building the container every time, you can just like define what you want in the container. And this kind of concept of just-in-time containers, which kind of flips the model on its head a little bit. Seqera containers really combines Wave with the concept of Conda and popular package managers to kind of provide that and make that all available as well. So Paula's going to do the kind of full demonstration and go through a little bit more of that after lunch. But really in a nutshell, why is it built? Why does it exist? It's really to remove that friction of users who want to create those containers. You can take those containers and then use them in Nextflow. You can use them really in kind of local development, if you want to run any notebooks, any really place you run the science. So it's a kind of a really open resource for people to use. Beyond containers, what about if we could benefit the same thing from pipelines? And sort of switching gears a little bit into pipelines, portability has really been a cornerstone of collaboration. When you think about the ability to like take a pipeline, you just get that experience. You just like Nextflow run and point to a Git repository. And when it works, it's amazing. And it's that kind of the feeling you get from that. I just remember it was the first time I did that. I was like, this is incredible. What we wanted to do is kind of like make that experience kind of available for everyone for all of the universe of Nextflow pipelines that exist out there. So we searched GitHub, found about 2,000 Git repositories would have some Nextflow files in them, which could consider to be thought of as a Nextflow pipeline. And we sort of wanted to understand like how many of those actually run and like can we make them available more generally. NF Core has done a great job of this. And the pipelines that exist there, they come with all of the things that you need just to run out of the box. But we wanted to understand like how could we make that more sort of widely available. There's a bigger universe of pipelines. Are you familiar with like Oxford Nanopore has got their Epitome pipelines, which are in Nextflow. There's also folks like the Sanger Institute, the Tree of Life. They've done a whole bunch of these pipelines. We wanted to kind of make that available, and that's why today we're also launching what's called Seqera pipelines, and this is available on Seqera.io. These pipelines are guaranteed to run in the sense that they come from a public GitHub repository. They all have the containers for the portability. They have test data sets which you can test, and we've been able to kind of go through and test all of those. And we manually created this list over the last couple of weeks, really to make sure that it was able to run and kind of launch in multiple places and run on that. Kind of thinking about it, it's really about finding and being able to run the right pipeline very quickly in your environment key, like with your data sets as well, and making sure that you have those test data sets. We plan to introduce a lot more automated quality metrics around this to really kind of expand that catalog, to have the community help, and really try and make this like the go-to resource for being able to find pipelines and kind of scale them out as well. So with that, I'm going to jump into a couple of demos here and kind of show you a little bit of how this works. Let's see if we can get some luck on this. Okay, that's a good start. Okay, the music would be cool to know. Okay, great. I think this is my first one here. I think I've lost a little bit on that one, but that's okay. Okay, awesome. So kicking things off here with Seqera Pipeline. So this is now available on Seqera.io, as I mentioned before. So from the homepage here, you can find pipelines at the top here. You have a bunch of different collections here. So for example, as I mentioned before, you've got like things like the epitome pipelines. You can find NF core pipelines that exist here. In my case, I'm going to kind of run through this example, and this kind of starts from the rare diseases pipeline. So if I just going to find that first, this pipeline it is basically taking whole genome sequence data or whole exome sequence data, running sort of variant analysis across that, and then doing ranking for that. So if I wanted to launch this now, I could got a couple of options. I can kind of go in through to select this. And we're using a really nice kind of mechanism here where you can either add the pipeline to a workspace. If you have an enterprise installation, you can choose your API endpoint, or you can also just launch these pipelines there directly with the CLI or with Nextflow or the NF core, kind of however you want to use your pipelines there. In my case here, I'm going to add this into my workspace. In this case, it's just going to be a Seqera Labs workspace, into the Boston Summit one here, and I can choose a compute environment. It's using a token to be able to get all of that information. And that's kind of like a really easy way. Eventually, we're going to like make this part of the authentication, which removes a whole bunch of that piece. So once I import this pipeline in here, I can then head over to Seqera Cloud, and it's going to pop up here. If I refresh this in a second, and we should find that that pipeline essentially gets added into there. It'll just take a couple of seconds to do, and it kind of pulls it in that we can run that. And you can see here now the rare diseases pipeline is in my workspace. If I want to launch that now, so I can go through and launch that pipeline, and from there you get the full sort of schema. This is coming from the Nextflow schema project, and we mentioned before a little bit about the stuff on the validation, but this enables you to have a full UI of that, which is available. Onto the pipeline itself. So we're going to spend some time kind of going through a little bit of this. The pipeline itself here, you can kind of get an overview. From here, you can see that in terms of NF core, you have the different stages of this pipeline. We're going to be running through this with just the default mode here. The input data for this is like FASTQ files, as well as some references. And they've done a very kind of similar thing as it relates to SRA Explorer in terms of pulling data in. So if I go to SRA Explorer here, you can see that I can search for that data. This is I'm going to be using the genome in a bottle data set here. So by searching for that, we can pull this up, and then we can basically add that directly into our workspace here. So finding the resource that we want publicly in terms of the pipeline. And you can see here now we've got this in our collection. I can select that data now and place that into my collection, which we can then export again into the workspace. So using a very similar mechanism here, you can see that we have our different samples. I believe in this case we've got three samples. They're split across 6 different things there, and I can pull them straight into that same workspace and launch from that. This provides me the kind of view of this data set here. Just give you a view of that. Here, I've added some other data sets, like the full data sets, which are coming from NF core. But just to give you a quick view of what this looks like, you end up having like the individual samples, as well as our pedigree here. So we know the maternal ID, the paternal ID, as well as the phenotype. So this is a kind of a typical trio study, which is being done there. If I wish to launch that pipeline, then I could take it in there, say, as I was before. I could say add some labels in there, but this gives me the ability to browse that, find the pipeline that we wish to run. In this case here, we're going to choose our test pedigree samples, and we can kick off that and launch that. Can show you a little bit of one which was run before, because this I think takes around half an hour or so, just even on the test data. And you can kind of see through the individual runs here. So I can jump into that one, and you kind of get a view of what's happening behind the scenes. Can track that and monitor everything as that goes through as well. So that kind of gives you an example of going from like a public resource, as I said, pulling it into Seqera platform, being able to find some public data, pull that data set in there. Often if you are going to do this, you may want to use something like fetch NGS to actually pull the original samples into your local, as opposed to running them directly from cloud, but it provides a kind of a nice overview of how to do that. Okay. So jumping back into the presentation now here. Okay, so far I kind of showed that how you could launch a pipeline. One of the key things though is when you start to run at scale and being able to kind of scale this efficiently. Scaling is not trivial, and particularly if you want to scale in an efficient way. You have many different variables that are at play here, different computer environments, different data sets. All these tasks require really huge different amounts of resources. Being able to do that in an efficient way is hard. So one of the things that we've spent really our 10 years now building out is the expertise and the kind of the ability to be able to right-size the tasks and build those computer environments in the efficient way. This is where something like Fusion comes in as well. This ability that you could mount the object storage inside versus kind of using traditional S3. We also have some optimizations around being able to select the right VM size and basically requesting the right amount of resources. And if we think about what our sort of customers and sort of users are saying about this, there's a couple of advantages. One is really kind of spending less. The kind of obvious one of just like the costs of running these pipelines at scale is much less. And we kind of get this when we see. There's also the reduction in manual effort. You don't want to have so much time spending on resource management, on people having to really kind of go through things in a manual way to do this, as well as that research productivity, like focusing on the science, using the software for better science and not doing that. So for us, as we've sort of built this out and have sort of advanced this, one of the things we also thought about, how could you just really focus on the science? And there is, of course, many ways that you can build computer environments, and I'll show you in a moment, but Seqera supports something like, I think, 12 or so of the different computer environments, Nextflow even more in terms of that. But how could you get away from that where you could just simply say, I want to run this in an optimized way? And this is where Seqera Compute comes in, and this is something that we are launching today in private preview. But Seqera Compute is this fully managed private computing environment. You spin up compute in the region of your choice, and really with that kind of one-click setup, you end up being able to connect your data, connect your public resources, and launch it in a way where you don't have to manage all of the things in the cloud, but still in a very scalable, efficient way to do that. A little bit more on that. It's really almost like an infrastructure as a service offering here. You can connect your data. You've got all the guardrails as it associates with that, and kind of do things, but it's still in a kind of very Nextflow-like way, where you're abstracting away the infrastructure, focusing on the science, and really kind of doing that. I can give you a very short demo of what that looks like as well. So this is available here. A little bit of a preview shot of what we see before. So in terms of the optimizations here, so this is in terms of the pipelines that we've just been running before. You can see here that I've got a pipeline which we're running like a default environment. This is just like standard S3. And on this pipeline here, it's like we have the same number of tasks there, so the 883. As I said, it takes about 42 minutes to run. Even on the test data set, we're using about 11 CPU hours, and kind of our rough costs on that, just of the task cost on compute is around 68 cents. And when we run the same thing there, kind of using an optimized way, so creating that computer environment, we can really save significant amounts just from not transferring the data around and sort of optimizing that here. You can see that we're down to about 4 CPU hours and sort of 16 cents on running the same data set through there. There's other optimizations that you can do on top of this. You can also take the existing runs that you have. So here's fetch NGS, and I can build a customized configuration. So I can take, for example, based on a previous execution, this is how much CPU I should request, this is how much memory I should request, and this config here can just be pasted in, just use it as it is, or also kind of just paste it in, and that can be very kind of adaptive and used in multiple places. So I mentioned before about these computer environments, and as I say, they have a lot of options that are associated with them. If you want to create, for example, an AWS batch option, you've got all the options in the world to do so. And for many customers, that's very useful. They have very specific requirements. For us, though, if you want to optimize this, you would simply select Wave, Fusion, and kind of build that out. What if you didn't want to manage that at all, though, and you wanted to have that direct? And this is really where Seqera Compute comes in. So inside Seqera Compute, you're able to create a compute environment, and simply by creating the compute environment, you have very few options in this here. So I would select Seqera Compute itself. I can just say, I'm just going to say Boston Compute. And here, I can simply select the region. This is important because you don't want to be transferring data, specifically across these different regions, so you can select that in there directly. And then once you have selected the region, you've got it in there that becomes available to you. It's fully transparent in the way that it builds with the same credentials. So it's got credentials which get created. It gets a bucket which is created for you as well. So when that bucket comes in, that's kind of where you're going to be using it for a working directory and pulling that through there. Cool, so that's a little bit on Seqera Compute. Now I'm going to jump into talking a little bit more around the data itself. And for us, that was what we built starting from about, I think it was Barcelona last year. We wanted to think about how we could make it much easier to not have to go into like object storage and go into like the cloud console just because you wanted to download data or upload data. How could we make it in a way where we could think about the data as almost like an abstraction over the existing primitives that people use, which for us is, object storage is the kind of the future of this, but in a way that still made sense for scientists and it wasn't like a lot sort of difficult to use. And Data Explorer really kind of came about to solve that problem. It provides this full visibility on your buckets, this ability to kind of link in buckets, but it also provides the ability to now have like public resources as well across different clouds, across different regions. And then you move the compute, you move like the workload to where the data is. And this is a big piece of kind of almost being cloud native and the way we think about all of those kind of public resources which exist there. So some of the new things that are launching today with Seqera Data Explorer is this multi-part upload, the ability to like upload files, download files directly within inside the browser, also using code, the ability to really collaborate on that, as well as to make it much easier to access data across different resources. And I'll just quickly kind of show you a demo on that one as well. Okay, so onto Data Explorer here. So here you can see in the same workspace that we were working before, I have a bunch of different resources. I've got like public resources. In this case, it's like the Dragon, these are 1,000 genomes which have been aligned with Dragon. And you can see that I can kind of get full visibility directly into a public bucket. So this is just with access into that. Supports for different cloud environments. I can also jump in and I can see things like, for example, the actual workspace that I was using for that data analysis that we've been doing before, which makes it very easy to do so. I can access in, for example, if I wanted to go find particular results that we had here. And I had, in this case, I'm going to kind of preempt a little bit of the work that we're going to be doing after this in terms of studios. But here I've got like the rank and the filtering, which is applied to all of those variants for those samples that we just ran before. So going back, this is really kind of using cloud storage to get the results of the pipeline that we've just run and be able to then take them, potentially share them if they wanted to. For folks that we're using here, you can download using code. The concept here is like still keep things easy for folks if you want to automate things, build things out in a very kind of large environment, but also the ability to like download them directly into the browser if that's what you want to do. I think one of the really cool features is when you're thinking about adding public resources in here. So for example, I can add a public bucket. I'm going to have to try and see if I can remember the name directly on this, but I believe that there is one we can add in the iGenomes, which is NGI iGenomes. I have to see the screen from a bit far away. But what that we're basically able to do is like load those into there, if this works. Yes. And then you can see that, for example, these are in EU West 1. Why is that relevant? Because it means when I create a compute environment, I can create a compute environment where those resources are and run my pipelines in that way, and I have the ability then to kind of browse on top of that. Okay, a few other things that you can do with Data Explorer. This is kind of constantly evolving as well, and hopefully you're going to see that. With one thing that I would point people to here is the AWS Open Data Exchange. It's got a really nice collection of about 500 different data sources that you can very easily link in, and that's something that we, in the future, are going to make it much easier to use inside of inside of the platform as well. Okay, now on to the final piece of the presentation that I have today, which is related to really the data analysis itself, and particularly the analysis, this part. We've focused a lot on pipelines, and that's a very hard problem, like very computationally intense problem. But how can we really do the things like either before the pipeline, data manipulation, data management, and after the pipeline where you're maybe doing some interactive analysis, generating figures? How can you do that in the same way that you develop an Nextflow pipeline? How can you do that with containers, with Git, in a way that you have very controlled environments that enable all of the kind of benefits that come from that? And again, in Barcelona last year, we released like the private preview of Seqera Data Studios. Now this is available today in public preview. What this provides you is the ability to spin up Jupyter environments, RStudio environments, VS Code, etc., in the same computer environment where you run your pipelines, and connected to the same data with the same collaboration features that exist as part of that. The goals here is really to make it really kind of like you don't have to leave the environment itself. You can have the output of a pipeline, which you can then instantly spin up a notebook in. You can then all host this still in your own environment. It's kind of running where you are, and just have it connected to those computer resources. Jumping onto the demo for this, and I would say this is probably going to be the most technically challenging part, but we'll see how it goes here. This is a list of the data studios. I've just previously, just before this, I've spun up some for RStudio, for VS Code. When you create a studio here, it's similar to adding a pipeline in. So you can choose existing templates. These are just containers. And you can add particular data in. You can see that you can run this in different computer environments. Now, the one I'm going to show you, I've created a computer environment with GPUs. And that's going to be relevant for some of the AlphaFold stuff I'll show you in a moment for basically using GPUs to be able to do that. You can mount data in, which is really interesting. So this is like allowing you to pull in, for example, different public data sets directly into that container and then be able to use them directly without having to move data around manually. And this is driven by Fusion. So inside of each individual container that gets spun up as part of a studio, it mounts that data, which just makes it really sort of easy to use there. Another feature that we've added in recently is the ability to have checkpointing. So every time a studio stops, it checkpoints the whole file system, and it checkpoints everything you've done with, like, for example, anything changes that you've made, any kind of additional software that you've built into there, so that you can go back to that at any moment. And then it also enables like collaboration. So if I, for example, have this studio, if someone else wants to come in and say, take over where I was before, they could take from the existing action here, create a new one, start from wherever that begins, and go from there. So that again provides this kind of collaboration features that are really kind of relevant. Let's go into this one here, and like this, I'm not sure on the Wi-Fi on this, but hopefully it will work here. So one of the things I've done here is I've spun up here, in this case, a VS Code environment. And the really relevant thing is that this is the same object storage from that pipeline that we just ran before. So I've mounted the output of that pipeline into here. If I go into here, I can kind of get some full visibility on that. I can go into, for example, and find the same data I downloaded before, the rank and filter stuff, and you can see that we've got that here. I've just extracted that VCF file so that we can view that. And if we were to, like, say, view that VCF file here, it's a little bit big, but hopefully it will open for us. We can kind of get that, pulling that directly from the object storage, and to view that with some luck. Let's just see, it's going to pull through. What I've got in that VCF file is just a ranked list of all of the variants that have come from that. What we could do there is, like, then, a typical workflow, maybe searching for the highest ranked ones, the ones that are going to have the most probability here for any kind of variance. So I'm just going to jump to the top here. And maybe a relatively simple analysis, but hopefully it illustrates the point here. If I search for, say, like protein encoding genes here, we can see that the first protein encoding variant that we find in this is in this TPTE gene. I could say, for example, either take that into something like PDB, do a search on that, What I've done sort of previously, I'd be able to like download that, pull that up, pull that into the studio that we're running. In this case here, I've got like a studio which I've just launched before. Hopefully this just takes a second. And here I've been running AlphaFold on top of that, so I'll be able to pull in the PDB file and provide, like, say, a viewer on top of that. Again, you can see that we're mounting the data so that we can kind of get full visibility on top of that. Just go to check the connection here. As well as then kind of running things in AlphaFold. What's nice about this one here is it like really runs directly the full kind of connection there. Just a little bit slower here. And you can see here I've put in the full protein, run through that. And it takes about half an hour or so to run on these Intel GPUs that I have there. But hopefully kind of shows you a little bit of the use cases that can exist there, going all the way from pulling a pipeline publicly, pulling in data from public resources, connecting them with your compute, with your data there, and being able to kind of go all the way through on top of that. Okay. So finishing things up, I'm just kind of like providing a little bit of a summary. Seqera Compute's now available today in private preview that you can use. We have Seqera Data Explorer, which is available as general availability today, as well as Data Studios, which is out in private preview. Summing things up here, so if we really think about sort of advancing science for everyone through software, as I say, this for us comes in through these three ways. Originally, we had, to start from the start of today, we had Nextflow. There's different areas, relates to building things, to discovering resources and scaling. Now with the new releases, we have containers, which help for discovery of new resources and pipelines. We've now added in compute, as well as Data Explorer and Data Studios, which are all available for folks to try out. A little bit of like a preview of what's coming up next. So as I say, I was really just scratching the surface of some of these things here. We have Paolo, who's going to be up and around 2:45 today. He's going to be talking about what's new in the Nextflow ecosystem and touching a little bit on going deeper into Seqera containers, as well as Vlad, who's going to be talking about multi-QC, some of the improvements that have been going on there, particularly as it relates to Plotly and some of the libraries underneath that, as well as finishing things off with Rob, who's going to be talking tomorrow and doing a much more kind of complete view of Data Studios as part of that. So with that, thank you all much for your attention, and yeah, be happy to take any questions. Appreciate it.
All right, thank you. That was a very exciting talk. It's really cool to see things getting into the public eye when they've been planned for such a long time. I'm personally very excited to be able to use eventually Seqera Compute for workshops and trainings to get that into the academics' hands as well. Anyway, we'll talk about that. There are some questions, just a few. For Seqera pipelines, why not just use TRS registry like Dockstore, which has supported Nextflow pipelines for quite a while?
Yeah, we looked into that. What we found was that let's say like the standard in terms of the actual pipelines that exist there just weren't able to launch directly. In terms of like supporting it as an API though, that's something that we can definitely do. The back end that we've built with Seqera pipelines as well as on containers can really kind of support any standards there, and that's something we'd definitely be willing to support there. It's really just kind of integration at the API level.
So to clarify, being able to support that as an additional option for bringing in pipelines?
Yeah, we could definitely support that on the back end. I would say the main focus for us has been actually the collection of pipelines themselves being high quality versus like the infrastructure behind that.
Yep. Fair. All right. Is there a way to transfer data between regions using Data Explorer?
Not directly via the UI, but you can always like go into a studio that you can bring up a console and do it in that way. It provides like a lot of flexibility around that. It's obviously a good idea if you're going to be reusing the data a lot, because the costs that are associated with transferring across regions can get relatively high.
And it seems like the mount data capability that you outlined for the data studios might help with that as well.
Yeah, absolutely, because you can mount different buckets across different regions there. I would say like if you don't have to move the data though, like it's a lot easier to kind of move the workload, move the compute if possible.
For data studios, are there some default pre-built containers, or do you have to build your own? And as a corollary, if there are some defaults, can you bring your own?
Yeah, great question. So the first one that we have in terms of the kind of launching with at the moment is VS Code, RStudio, as well as Jupyter. The vision for this, though, is to have very much a similar interface that you have with containers, with Seqera containers, to be able to really add your own packages into there and then kind of spin those up as studios. There is ways to build custom ones today. It's a little bit, still a little bit hacky at the moment in terms of like what we're building, because we want to create that as really a kind of proper product feature, and that's on the roadmap for like coming up very shortly.
Awesome. Well, thank you. That's it for the questions that we have now. So thank you very much, Evan, for this very exciting talk. We'll look forward to the additional details about some of what's coming.